Welcome students to this module of e-learning. I am Dr. Maria A. D'Souza, Assistant Professor, St. Xavier's College, will present to this module to the third year students of Botany, Semester 6, Course Code BOC 109 and the course title Molecular Biology and Genetic Engineering. The module name DNA Sequencing, Maxam and Gilbert's Method, and the title of the unit Recombinant DNA Technology. The outline of the module, students, is principle of Maxam and Gilbert's DNA sequencing, the requirements, procedure, the advantages and disadvantages. The learning outcomes. Once the student completes this module, the student will be able to explain the basis of Maxam and Gilbert's method of DNA sequencing, describe the steps involved, and the advantages and disadvantages. So students, to begin with, today you are going to do Maxam and Gilbert's method of DNA sequencing. This method was put forth at the same time when Sanger also did its sequencing method in the year 1976 and 77. Alan Maxam and Walter Gilbert were the two scientists who proposed this method of sequencing. It is a method by which the sequence of a double-stranded or single-stranded DNA fragment is determined by treatment using chemicals. Now, these chemicals cut the DNA at specific nucleotide position. So, it involves the chemical degradation method of DNA sequencing. It's also called by that name, students, because it involves the reaction in two stages. Number one, involves the chemical modification of the bases in DNA.' modified base is removed from its sugar. That means piperidine cleaves the phosphodiester bonds 5-N and 3-N and the base is released. Now, the steps involved in the chemical degradation method or the Maxam and Gilbert's method. First, the double-stranded fragment is separated into two single strands, either by applying high temperature or treating with high pH, that is treating with NaOH. Now, these fragments are run in a gel so that you separate them out into fragments. These fragments are radioactively labeled, students. And which end? It is the 5-N and they are labeled with a phosphate enzymatically, 32PO4. Once you radioactively label these fragments, place them in 4 test tubes. Now, these 4 test tubes will be subjected to the chemical cleavage. These 4 test tube number 1, the cleavage will occur at wherever there is A plus A and G, G only, T and C, and C only. So, for this specific chemicals are added. In test tube number 1, we have formic acid and piperidine. Test tube number 2, we have dimethyl sulfate and HCl. And hydrazine in the third one and hydrazine and alkali in the fourth one. Now, piperidine is added to all the 4 test tubes because it will cleave the phosphodiester bond at the 5-N and they will release the base. So, now, if we see the 4 test tube, in test tube number 1, where formic acid was added, it will cleave wherever there is guanine and adenine. So, if you see the strands, they are separated at places where there is adenine and guanine. Dimethyl sulfate will cleave wherever there is guanine, whereas hydrazine will cleave the fragments wherever there is cytosine and thymine. Whereas hydrazine and alkali in the test tube number 4 will cleave only at cytosine. So, you will see that the chain breakage occurs at a specific nucleotides, whichever chemical is added. Reaction proceeds long enough to produce an average of 1 break per strand. Now, these random breaks generate end label fragments, representing all positions of each indicated base. Now, these labeled subfragments created by the 4 reactions have at one end radioactively labeled P32, 5-N. And the other end, they have the chemical cleavage point whichever chemical is added, whether there's C, T, G, A, whichever, they will cleave at that point. because of the particular chemical added. Now, reaction products are separated after that by polyacrylamide gel, wherein each test tube is put in a particular lane or in a particular slot. Now, these label fragments are later on, they are subjected to gel electrophoresis, wherein they are separated based on their sizes, molecular weights, and then they are visualized by autoradiography. And then we read the sequence from bottom to the top. Now, this is what you see, students, is an autoradiogram, but this is through Maxam and Gilbert's method. Now, if you see the separation wherein formic acid was added, and this is where DMS, dimethyl sulfate, was added. If you see a band like this in both the places, how do you read the sequence? This is G and A, and this is G. So, G is repeating in both, so you write a G. So, then accordingly, C, A, this is repeating C plus T, and this is T, so T. So, this is how you write your sequence. And the sequence, students, is read from bottom to the top. That means you will read it from G, C, A, T, T, A, G, C, T, A. So, this is how you will get your sequence from the fragments which are cleaved by the chemical reaction. The advantages of this method, students, it directly reads purified DNA rather than how you use for Sanger's method. It will not make a copy of it. It will cut the original fragment of DNA at the specified base because of the use of chemicals. Moreover, there is no premature termination due to DNA sequencing. So, no problem with polymerase to synthesize DNA. Stretches of DNA can be sequenced, which cannot be done with enzymatic method. And it is also used, students, to analyze DNA-protein interaction, epigenetic modification, and nucleic acid structure. So, these are the advantages, students. There are also a number of disadvantages, students. This technique is not widely used like Sanger's because the setup is quite complex. There is extensive use of toxic chemicals, radioactive isotope. You saw that PO432 was added, radioactively labeled at the 5' end. And these chemicals and these radioactive isotopes are very poisonous and unstable. You cannot read a sequence more than 500 base pairs. This one. And it is very difficult, students, to make a Maxam Gilbert DNA sequencing kit. And the read size decreases with incomplete cleavage reactions. So, you see, students, there are advantages, but there are equal number of disadvantages. So, because of this reason, Sanger's method, students, is more used than Maxam Gilbert. So, these are the references, students, offline references. And these are the online references where you can see how the procedure is carried out. And thank you, students, for a patient hearing.